So it is going to be all about paper 2 today. We're looking at some of the math skills and some of the questions that are frequently assessed as well. And you can get this booklet for free. You just need to go to missestro.co.uk/paper 2. So that is where you can get that from. So I'm going to zoom this out a little bit, which will make it a bit better for viewing on Instagram. I think it was a little bit too zoomed in. Okay, so it would be really helpful if you are here from the start all the way through. Can you do me a massive favour, please? Because I'm not really looking at the chat box too much because, as you can see, I'm filming what I'm actually answering. So if anyone asks any of these questions, here are the answers, and please can you share it? Number 1, yes, it is being recorded and it is being saved on both my Instagram and YouTube channel. Number 2, this is AQA, A-level biology, paper 2. Number 3, you can get this booklet for free. It will be linked at the top of every page. Those are the 3 most commonly asked questions that I get in all of these lives. So if anyone does ask them and you are here throughout, then yes, please tell me it is recorded on Instagram and YouTube. Here's how to get the papers, and it's AQA A-level biology. But let's get started. I'm going to double check that each platform can see okay. I think so. So first thing I just wanted to share with you before we do some questions is some little tips for paper 2. I have actually also linked, I do my pick and mix live lessons. If you didn't know, those are the paid ones because you get the slides. It's like an hour of tuition with me. I answer your questions, whereas I don't really do that in the live, so I'm just focusing on this. The gene technologies one is completely sold out already, so you can't sign up to that one anymore. That's tomorrow evening. The recording will become available, but the paper 2 comprehension one, there's availability. So this is the night before the exam at 8 p.m. If you download this document, you'll get the link, but also just comment pick on my Instagram on any post and you'll get the link as well. So that's just what this information is at the top. But here then, you might have seen my YouTube video or some of the short form videos that I've done on this, where I've talked about commonly assessed topics and therefore what may or may not come up. And as I always emphasize, these are not predictions. Nowhere have I ever said predictions. It is just data analysis, commonly assessed topics compared to what didn't come up in 2024. And then it gives you a combined list. And for context, the OCR and the AQA one for paper 1, from what people have told me, I've not seen the papers, they told me it was spot on. The things that did come up on the commonly assessed one and weren't on the 2024 paper, all of those were assessed on those papers. So that's why I thought I would just highlight it again here. As I said, doesn't mean it's predictions, doesn't mean it's definitely coming up on paper 2, but it's just worth being aware of that. So here are 4 commonly assessed topics, and this just tells you what percentage of the time those have been assessed. So we can see the most common ones are nervous coordination, homeostasis, gene inheritance and gene technologies. One thing just to raise here though, because it might be surprising, you're thinking, well, why aren't photosynthesis or respiration on there? The reason for that is it's normally one or the other. So photosynthesis and respiration, if you consider those together, I think it's actually close to 100% of the time you either get photosynthesis or respiration. So you could always consider which was focused on more heavily last year. I think both of them came up a little bit last year, but I'd add this to the list of ones to make sure you definitely know. In particular, the photosynthesis required practical, which is required practical 8, that's not been assessed in a long time. So there's potential that maybe photosynthesis might be linked to that required practical 8, the one with the DC PIP. Again, not predictions, it's just based on what hasn't been assessed in a while. So respiration, I think respirometers came up in 2024, but again, not in loads and loads of detail, and sometimes one of them's assessed on paper 2 and the other's on paper 3. So basically, I would learn those. But the big ones that do stand out are nervous coordination, which basically means the nervous system, and gene expression are ones that are commonly assessed and didn't come up last year. Having said that, gene technology seems to feature in some way or another basically every year. And the homeostasis, that is obviously a very broad scope there. It doesn't tell you whether it's diabetes, blood glucose control, or water retention in the kidneys. But it was just to give you a bit of an idea, because I know a lot of you have been panicking since paper 1. I still haven't looked at it yet. I know a lot of people have asked me, have you looked at paper 1 yet? Not yet. I do actually have a copy, but I've been in, you might have seen, I've been in Seville. I landed today and I haven't had a chance to look at the paper. I've been too busy getting ready for everything for paper 2. So I will have a look at it at some point and give a bit more feedback, especially then when I've seen paper 2, because that means I'll then reassess my data analysis to see what came up on paper 1 and paper 2, to then try and consider what is really the most likely thing to come up on paper 3 and potentially the essay. So yeah, if you did want this document, it is completely free for you to download, missesther.co.uk/paper 2. So you've got like this intro page here going through that information. Then the rest of it is the exam questions that we are now going to have a go at. So fur colour in cats is controlled by 2 genes. One gene is sex-linked and located on the X chromosome. So that's quite a common question to do with like sex linkage, is fur colour in cats, because you can get orange fur, black fur, and if it's codominance, then that means both of them are expressed. Inheritance did come up last year though, so potentially not in a lot of detail this year. The alleles that code for the colour, pigment or codominance. The allele G codes for ginger fur and B codes for black. And we're told that tortoiseshell is only possible in females because it's codominance, it's sex-linked, which means it's on the X chromosome, and only females would have those 2 X chromosomes. A second gene, which isn't sex-linked, can mask the expression of the gene for fur colour. There are 2 alleles for this gene. So let's get this here. So 2 alleles for that gene. F is dominant, and if it's present, then they don't make any pigment and the cat will be white. And if it's recessive, they do get the pigment. So the first question is, name the type of inheritance of that F gene, of the interaction between those 2 genes affecting fur colour. So if we're told that one of them prevents the formation of that, it's basically affecting or masking, and that is epistasis. The next thing then we've got is, what would the phenotype be of these? So we've got, first of all, it's a female, and just to make you aware, whenever they do give you the sex chromosomes, part of the phenotype is actually always stating the biological sex as well. So this would be female, and this is male. So we've got G and B, which would mean it's tortoiseshell. Let's just double check though. 2 recessives, and it was dominant that makes them white. So it would be a female that is tortoiseshell. I have to pause to check the spelling. Right, then we've got our male, which is G for ginger. They can't be tortoiseshell because they only have 1 X chromosome. They do have a capital F, which makes them white. So that is a white male cat. Okay, then we have the genetic diagram below, a dihybrid cross. We have to say the genotypes and phenotypes of the offspring and the ratios. So first of all then, when working out our gametes in a dihybrid cross, you always have to have 1 of each gene in a gamete. So for example, I'm going to do this parent down this side. We'd have X, capital G, capital F. That's 1 potential gamete. And I always do my gametes in a circle just because it avoids any confusion between the genotypes. You don't have to do that, but it is like a convention that is done. X, capital G, lowercase f is the other. So that's our first parent. Then this parent, X, capital B, lowercase f, or Y, lowercase f. So now I need to combine those. And then my last one. Now, there is a reason why I've deliberately done this higher up in the box. It's because I want to leave space to write in the phenotypes, because they do want you in mark schemes to match the genotype to the phenotype. And you could do that by either writing it all out again here and making sure you've got it matched underneath. But I'd save myself time in the exam. You don't literally have to write it here. You could just do an arrow and say, like, these are my genotypes. You don't have to write things twice, save yourself time. So this is a tortoiseshell. Well, actually, hold up. We've got a capital F, so it's white, 2 X's, female. So that is white, female. This is male, capital F, so that's white. So white male. We don't have any other capitals now, so this is a tortoiseshell female. And this is an orange, or did they call it ginger? Ginger, okay. Sometimes I say orange, sometimes I say ginger. Ginger, male. So that means my ratio is 1 to 1 to 1 to 1, because you do have to consider the sex within that phenotype. So you wouldn't say 2 white to 1 tortoiseshell to 1 ginger. That is a different phenotype. So we've got 1 to 1 to 1 to 1. And I don't need to match it up because it's all the same ratio. They're all to 1. So that's why I haven't matched it again. Right, last bit of this question is a Hardy-Weinberg. So we've got 43% of cats have the F allele. Use the Hardy-Weinberg equation to calculate the frequency of the lowercase. So although they've said F allele here, which is the dominant, that doesn't mean that they have given us the letter P, because P is the frequency of the dominant allele, but that's not what they've said. They haven't said the frequency of that allele is 43%. They said 43% of the population have that allele. So if they have it, then that means 43% is actually either the homozygous dominants who have that allele or the heterozygous individuals. So 43% is P squared plus 2PQ, but we've got 2 unknowns there. So if ever they give you the frequency of the dominant genotype, then, or dominant phenotypes, by the way, because they're 2 genotypes, then I would then just say, well, what is Q squared then? Because if we remember, P squared plus 2PQ plus Q squared equals 1. So this is 100%, which means 57% is what Q squared is. So if Q squared is 57%, that means 0.57. So Q is the square root of 0.57. So Q equals 0.75. But hang on, I've jumped ahead of myself here, not even looked at what I need to work out. So use the Hardy-Weinberg equation to calculate the frequency of the F allele. Oh, and that is what Q is. So we've actually got to the answer already. They just want to know what Q is, because Q is the recessive allele, and they want the frequency of that. So that is our final answer, 0.75. Oh, I didn't actually check my rounding of that, but double check it. Let's just double check that again. .75. Okay, yes, so that is fine. And if they don't say that they want it as a percentage, that means they'll accept the frequency as 0.75 or as 75%. But just read questions carefully, because sometimes they explicitly say as a percentage, and then you would have to times by 100. Okay, next question. Energy in ecosystems, energy transfers hasn't come up in a while, and calorimeters has only really come up once, so I thought we should have a look at this just in case. So chemical energy stored in biomass is measured using a calorimeter, not to be confused with colorimeter, very different. So if you haven't come across this, that is what it does. Chemical energy stored in biomass can be measured using a calorimeter. And what they do is put a known mass of whatever biomass sample you're using, completely burn it inside of this calorimeter, and they measure the start temperature of this water surrounding it, measure the end temperature, and look at the temperature increase, because that can be used as a measure of how much energy there was, chemical energy in the biomass, because as that burns, it releases heat energy, and that heats up the water. I think it is in chemistry as well. So looking at the diagram, explain the purpose of the stirrer and the layer of air around the outside. Okay, so that's our 2 questions here. So the stirrer for this one, that is going to help to distribute the heat energy, because if our thermometer is only here, we do need to make sure any heat energy that's being released from the combustion is being equally distributed. So that's what I'd go for here. And then the other bit was this layer of air around the outside. Air is an insulator, so that's going to reduce any heat being lost to the surroundings. It's all going to be going to that water instead. So I'm going to say air acts as an insulating layer. Okay, let's see. Next, going on to another maths question, because I did say I was going to focus it quite heavily on maths, because a lot of people said they liked the maths skill one that I did for paper 1. So I'm kind of doing a mixture of maths and theory. So for this one, let's have a read through. So we've got a 3-gram sample of biomass fully combusted in the calorimeter. The volume of water in the calorimeter was 200 centimetres cubed. The increase in temperature recorded was 16.7 degrees C. We're told that 4.18 joules of energy are needed to increase the temperature of 1 centimetres cubed of water by 1 degree. Uses information to calculate heat energy released in kilograms per gram of biomass. Now, you can do this using the formula that you learn in chemistry, but if you don't take chemistry, you don't have to know that formula. You can still work it out using this information. By the way, you might, if you were at the last math session, I did say I don't like how in the mark schemes, they just tell you the final answer. They don't give you all the workings as you go. So if you do download this for free, you get all of these maths workings, because I realise that sometimes just being told the final answer isn't very helpful. So what we need to do here is calculate the total energy released in joules first of all. So to do that, I need to do my mass of water, or basically that is volume, times specific heat capacity times the temperature change. So that is 200 times 4.18 times 16.7, and that comes to so that's how much energy. But we've got it in joules still, and they want the answer in kilojoules. So we now need to convert that into kilojoules, which is 1,000 times bigger, that unit. So we need to divide this by 1,000 to convert it into kilojoules, sorry. So into kilojoules, that would be divided by 1,000. So 1, 2, 3. So we actually have 13.9612 kilojoules of energy, but we need to do that per gram. So we are doing this per gram, and we've got a 3-gram sample. So the last step to make it per gram is divide this answer by 3, which comes to 4.6537. Have they told us what they want it to be rounded to? No, they haven't. So I'd do it to 2 decimal places. I'd say 4.65 kilojoules per gram. Right, next question. We've got another just random maths question now, similar to one of the concepts that came up in the last one we did where it was looking at cell division. So algae cells can divide rapidly. A culture of 500 cells was set up in a fermenter and the cells divided every 120 minutes. You can assume that there were no limiting factors, no cells died during the 24 hours. Calculate the number of cells in the culture after 24 hours. So 24 hours, they divide every 120 minutes. So the first thing I'm going to do is convert 24 hours into minutes. So I need to times that by 60. So that's how many minutes we've got. And if the cell divides every 120 minutes, what I'm then going to do is divide that time by 120 to see how many divisions we get. And that is 12 divisions. Now we can use the formula that I've talked about quite a bit. Whenever you need to know that, so I was just getting distracted by some of the questions coming up on Instagram. Oh, someone's asking why was it divided by 3? Yeah, that was this. So you don't always divide by 3. It's because I wanted it per gram and it was a 3 gram sample. Anyway, back to this one. Yeah, whenever you need to know how many cells you get at the end after divisions, because when something divides, it's splitting in half. You get 2. It's always 2 to the power of n, where n is the number of times it divides. So for this one, that is 2 to the power of 12. But this is going to happen for all 500 cells present. So if it's happening for all 500, it's 2 to the power of 12, but that is happening 500 times, which is why we have to do 2 to the power of 12 times 500. So 2 to the power of 12 is 4,096. Need to times that by 500. And that comes to 2,048,000. But I think they asked, yeah, they wanted it in standard form. So in standard form, 2 point, they haven't said how many decimal places, but I'm going to go to 3 significant figures. So that's what I'd have as this answer here. Okay, right, final question today, everyone, is, again, because I said energy and ecosystems, energy transfers hasn't come up a lot recently. To be fair, it doesn't get assessed a lot, but I just wanted to remind you from like a sort of maths point of view, but also theory, there is this equation as well. So let's have a go at this into the chat box. Which of these, 1, 2, 3, or 4, is the correct formula for net production from consumers? And then we'll say what the formula is for GPP. I'm probably going to see answers coming in quickest on that one. So I think everyone's saying number 2 that I can see. On TikTok, everyone's saying number 2. So let's have a look. So energy is what is being ingested minus what is lost in the feces and respiration combined. So yeah, it is that one. Then what is GPP? So you learn on the spec the formula for NPP. So if they do say to you, what is NPP or what is GPP, and it's 1 mark, the formula is sufficient. You don't have to write a descriptive definition. The formula is sufficient. So what is GPP? You just need to give the formula here for what gross primary productivity is. And as a reminder, on the spec, you actually are told NPP is GPP minus R. So that means GPP is NPP plus R. That would get you the mark. So you do not have to write out a wordy definition if they say what is GPP or what is NPP. And just as a little top tip, if there is any question, an equation, I'm losing my words. If there is ever a question where it links to either GPP or NPP, just put the formula in anyway, because the chances are that is worth a mark anyway. I can also see some people asking what I, F and R stand for, so I'll just write that here. So that is ingested, feces, respiration. So like energy input from the ingested, energy lost in the feces, energy lost in respiration. And N is the net production of the consumers. Okay, so that is it for the paper 2 live. I think it's probably the last one I'm doing on social media before paper 2, because I am doing my gene technology pick a mix lesson tomorrow evening, but that one's sold out. I will make the recording available probably about an hour afterwards, because I've had so many people request can they still come, but it is sold out. There's a cap on that one. There can't be more than a set number. But the other one, I'll just go back to this first page. The other session, which is Thursday, the night before the exam at 8 p.m, that one is still open. Either comment on any of my Instagram posts, pick, so literally PICK, and you'll get the link. Or if you are downloading this booklet anyway and you want to come along, then you just need to click here to get that link. But that is it for today's live. Hopefully you found that helpful. I do have a final YouTube video coming out. I think it's tomorrow, with last minute advice for paper 2. So make sure you watch that for any last minute advice as well.